I'm showing that to you because I want to show you other things that can go wrong, which might actually lead to side effects and unexpected events. When you want to get that gene construct into the plant, that's what we call transformation. The transformation process, how do you get it in there? For example, in this case, for SmartStax, we had agrobacterium use, which is one of the shuttle mechanisms based on bacterial background to get a gene into a cell. The black part is sort of like the DNA I'm posting there. And then another mechanism is the particle bombardment, where you shoot them in on gold particles. Both these methodologies have been used in SmartStax. Different lines have used different transformation mechanisms. Now, as a result, you can see they end up in all different places. You have a random integration, imprecision, and actually what we noticed when my group was looking at the different rates of mutations, one has hundreds to thousands of mutations from both that process as well as the transformation process of making actually plant cells, turning them, making them grow into plants again. So that is one of the papers we've been doing, and if you look at that, you can see that the effects of the transformation process is something what we call genome scrambling. At the insertion side of the genes, you can have very, very strong alterations, deletions, turnarounds of DNA. They have been moved around in the plant genome, or even if you look wider, you have lots of injury components in the plant where you have lots of point mutations, which might not be of any consequence because mutations occur, but then, having worked in the field of hemophilia and gene therapy, I know that one point mutation is enough in order to make the whole gene unfunctional. So one never knows, so it needs strict assessment, which means we should not just look at trait, as I said there, but we need to really look at the whole plant. And when you do the stacking, of course, you bring all these backgrounds, these injuries, into the new set. And it might be fine in one of the original lines, but if you add them together, you might have all of a sudden effect you just had not anticipated at all. Most genes interact and work in combination with others. It's just a very, very few genes that just work on their own. So basically, we have an interaction, and we also have something which is multi-trait genes, or rather I don't know what to call it. A trait that requires quite a number of genes in order to produce one trait, like often drought resistance is one of those. Or for example, like a number of vitamins in the production need quite a number of genes involved in it. Or for example, the cholera toxin, which is based on two different proteins, on the A protein and the B, and you need 1 unit of A and 5 of B. They've already done some genetic modification, I think, of plants with the B subunit. But so basically, if you assess a plant, in this case with the cholera toxin as an example, with the cholera toxin A, and another plant with the cholera subunit toxin B, you might find that there is not a problem. But that would never make you think that when you put it together, that you wouldn't have to assess it again, because you know you have started off with a toxin, or something that could build a toxin. But that would mean if we only assess, if we know there is a combinatorial effect, then we would miss out the ones which we do not know. But by that, we actually already acknowledge that it's not sufficient to just assess the parental lines and not the combination, because that is one of these cases where it's quite clear that we have to assess it. I will not go through much more of this. The choice of promoter, we could look at, because in this case, for example, gene silencing means that there is a switch-off system. Can I have a few more minutes? Thanks. Gene silencing is a mechanism that can basically switch genes off, but not necessarily forever. It can be switched on later again, so that is not heritable. It's something which is amongst the mechanisms which we call epigenetics. But quite often what we found is these transgenes that get switched off more often, especially under environmental stress. But often what is also happening is if there is repetitions of sequences. And I've looked at the different components in this SmartStax, and it's quite extraordinary, like that the promoter sequences of the 35S promoter, the on and off switch, or in this case it's just an on switch, it's there 4 times, right? So therefore, we don't know how long we can actually rely on the stability of that, or that the system will actually function properly. Therefore, it would need to be assessed over time under environmental stress conditions in order to know, will it switch off or will it not switch off. Another problem we have when we have used homology sequences, like if we use the same promoter, is that then we might actually invite in the reproductive process a crossover, which seems to be a homologous recombination, but actually they would be in different areas of the genome, and we would have all of a sudden crossovers that are creating a completely altered genome with different characteristics. So, but that hasn't been assessed either. If you look at the different sequences, it's quite interesting to study them. My conclusion is that SmartStax has to be assessed as a new GM event. There is no way about it, with particular emphasis also on combinatorial and synergistic and cumulative effects. Another point, a toxicological rule, or a rule like which is used in toxicology, is like if A is safe, if you feed an animal with a certain component, with A, and it's safe, and then you feed an animal another lot with a substance B, and that is safe. That does not mean if you feed a combination of A and B to animals, that that is safe. Because actually that is quite often where toxicological effects of chemical interaction take place. And that's where in Europe we have a vast reassessment of chemicals going on now because of these impacts, toxicological impacts of synergistic effects. And finally, assumption-based risk assessment or decision-making is, for one thing, unscientific and puts health and environment and agricultural performances at risk. Therefore, I feel we have the obligation as scientists to make the risk assessment as strict as possible and to make sure that we assess it properly and that assumption-based risk assessment is clearly not done, that we say assumption-based risk assessment has no place in assessing GMOs. Thank you.
So what I'm going to try to talk about fairly quickly is first go through this issue of codex. What does codex actually say about what you might call stack traits and whether they should or could be go through a required safety assessment. Then I'll talk about some unexpected effects that can happen following on what Ricarda said, and we'll also talk about potential health problems, particularly the issue of allergenicity with the cry proteins, and then we'll end with some information about what the EPA is doing in this area. So if we look at Codex Alimentarius, that's the Food Safety Standards Setting Organization of the UN. It's a joint WHO and FAO. It was set up in the 1960s to help developing countries with a range of food standards that were voluntary food standards, guidelines, and recommendations associated with food safety. This was something that countries could take and do anything that they wanted with these recommendations. None of them were legally binding.